Welcome, curious minds, to another deep dive. For centuries, the Black Death has been, well, this terrifying specter in our history, hasn't it? Its origins, its spread, kind of shrouded in myth and fear. I mean, imagine a single disease so devastating it claimed maybe up to 50% of Europe's population in just a few short years. It's this huge historical mystery that's haunted us. But today, we're going to pull back that veil, using, the sharpest tools of modern science to plunge into this ancient enigma, and maybe even rewrite history itself a bit.
That's absolutely right. For the longest time, the exact nature of these ancient pandemics and, well, precisely how they relate to the plague we understand today, that was all wrapped up in some pretty fierce academic debate. So our mission in this deep dive is really to go far beyond the textbook stuff. We're going to explore how the incredible power of ancient DNA, or aDNA as it's called, coupled with some really advanced genomic analysis, is definitively confirming Yersinia pestis as the agent behind all of this, across these huge stretches of time. And more than that, it's revealing its incredible journey through history, its sort of evolutionary twists, and its really profound impact on human biology.
Absolutely fascinating, because this isn't just about understanding some plague from the past, is it? It's about seeing how it shaped us, maybe right down to our very genes, and why this seemingly ancient threat is still, well, incredibly relevant in our world today. So if you've ever wondered how scientists can reconstruct the full genetic blueprint of a pathogen from centuries ago, or how our own bodies might have adapted, maybe evolved in the face of something so catastrophic, then you, our curious listener, you are in for some truly surprising facts and probably more than a few aha moments. Consider this your shortcut to being well-informed on a topic that really blends history, cutting-edge biology, and I guess the sheer resilience of life. Let's unpack this and unravel the full story of the Black Death, from the ancient shadows right up to its contemporary presence. Okay, let's start with the shadow of the past, because plague isn't just some grim historical footnote, right? It's been a recurrent, devastating threat throughout human history, striking in these three major waves that literally reshaped civilizations, altered the course of human development.
Yeah, what's truly fascinating here is just the sheer scale and the recurrence of these events. It reminds us that infectious diseases are definitely not new challenges. While the Black Death is probably the most famous one, the first known major pandemic was actually the plague of Justinian that ravaged the Eastern Roman Empire between the 6th and 8th centuries AD. Its impact was, well, profound. It contributed to the decline of cities, destabilized trade, and really altered the whole geopolitical landscape of its time. It's a sobering reminder of plague's capacity to undermine even powerful empires.
And then, of course, came the Second Plague pandemic. This spans from, the mid-14th century all the way through to the mid-18th century. And when we talk about the Black Death specifically, that initial, most devastating wave from 1347 to 1353 AD, the mortality rate was just staggering. Can you even imagine half the people half the population of Europe, wiped out in just a few short years? It's almost incomprehensible, isn't it?
It genuinely is. The prevailing historical thought, and this is now really bolstered by genetic evidence, is that it emerged from these persistent plague foci in East Asia, likely what we call natural reservoirs in wild rodent populations. From there, it began its inexorable journey west along the ancient trade routes. We have pretty compelling historical reports detailing its arrival in Messina, Sicily, in October 1347, carried by Genoese ships. These vessels, tragically, were laden with flea-infested rats, which were just perfect biological vectors for the disease. From Sicily, the plague spread rapidly, incredibly rapidly, across the entire European continent. By 1348, it had engulfed France and Spain. A year later, 1349, it was rampaging through Germany, Switzerland, Austria, and then onward into Scandinavia. Finally, by 1351, it had arrived in Russia, coming in via the Baltic coast. Its speed of spread was truly terrifying, especially given, the lack of understanding about how it was transmitted.
It sounds like it just swept over everything, like a relentless tide. Were there any places that were, I don't know, initially spared from this first devastating wave? It seems almost impossible.
Yeah, there were actually a few interesting exceptions. Iceland, for example, remained largely untouched by that initial Black Death wave until 1402. But when Yersinia pestis finally did arrive on its shores, it caused an equally horrific decline. Some estimates suggest a 50 to 60% mortality rate in just 2 years. What's also really critically important for our understanding is to remember that the Black Death wasn't a single isolated event. Plague continued to strike Europe in subsequent waves for centuries after that initial catastrophic period. This ongoing series of outbreaks constitutes what we call the Second Plague Pandemic. It was essentially a centuries-long nightmare.
And then, much later, came the third plague pandemic, late 19th century. This one started in China in the 1850s, but it became globally significant, didn't it? Marking a whole new chapter in plague's history.
Yes, profoundly so. It originated in the Yunnan province of China, and its spread was, well, tragically facilitated by various socioeconomic factors, notably the Panthay Rebellion, which caused this diaspora of refugees who unfortunately carried the disease throughout China. From there, British merchants, who were integral to the global trade networks of the time, they transported the pathogen from Hong Kong to other parts of their vast empire. India suffered the most substantial losses, losing millions to the disease. However, this era was crucial for entirely different reasons. It was during this third pandemic, particularly in Hong Kong, that these groundbreaking scientific advancements occurred. Scientists definitively identified the causative agent as the Yersinia pestis bacterium. And crucially, they figured out the complex relationship between rats, fleas, and human disease transmission. This marked a huge turning point in our understanding of infectious diseases globally.
So, given these three massive, distinct waves of devastation, separated by centuries and geography, there was a significant historical debate, right, about whether they were all caused by the exact same agent. Because the mortality rates and, the epidemiology sometimes varied. Was it truly the same killer every time?
Precisely. For a long time, there was genuine controversy over whether these distinct pandemics shared a common cause, a common etiologic agent. Some scholars argued for different pathogens or maybe entirely different modes of transmission. However, recent years have seen a pretty definitive resolution to this debate, thanks almost entirely to the remarkable capabilities of ancient DNA or aDNA analysis. It has now been definitively confirmed through pretty much irrefutable genetic evidence that Yersinia pestis was the responsible agent in both the Justinian plague and the Second Plague pandemic, including, of course, the Black Death. This scientific evidence has really clarified a long-standing historical question, kind of uniting these disparate historical calamities under a single biological cause.
Okay, so we know Yersinia pestis was the culprit across centuries, but how did this, seemingly unassuming bacterium become such a master of adaptation? How did it transform from a relatively harmless organism into one of the most virulent human pathogens we know? What was its evolutionary leap?
That's where its remarkable evolutionary leap comes in. Yersinia pestis didn't just appear out of nowhere as this highly virulent killer. It evolves from a less virulent, closely related cousin called Yersinia pseudotuberculosis. This transformation involved acquiring specific genes and plasmids, which are like these tiny, independent mini-chromosomes that bacteria can swap and pick up, that conferred upon it incredible new abilities, basically turning it into a truly dangerous pathogen. What's even more fascinating, and this is a relatively recent discovery thanks to ancient DNA, is that its early presence has been identified in ancient human material from Altai, Siberia, dating back as far as 5,000 years ago. However, at that very early stage, it likely didn't have the full genetic toolkit, particularly the ability for efficient flea-borne transmission, which is what typically leads to those massive outbreaks of bubonic plague. That ability seems to have been a later evolutionary development that truly unleashed its pandemic potential on a global scale. It's really a masterclass in bacterial evolution.
So it gained these abilities almost like a genetic arsenal, piece by piece, over millennia. Can you tell us more about these key virulence factors, these specific tools that make Y. pestis so incredibly effective at causing disease and spreading?
Absolutely. The crucial role in its extreme virulence is played by three well-characterized plasmids. As I mentioned, these are small, circular DNA molecules,
Hmm.
separate from the main bacterial chromosome. These are PCD1, PBCP1, and PMT1. Think of them as individual powerhouses, each encoding many of its most potent weapons against a host's immune system.
Let's start with one that seems pretty central, the play gene. What does that do, and why is it so important?
Okay, the play gene. It's located on the PPCP1 plasmid, and it encodes something called the plasminogen activator protease. This protease is incredibly important for the bacterium's ability to invade host tissues. It works partly by degrading fibrin clots, allowing it to move rapidly throughout the body and establish a systemic infection. It's also a significant factor in lung infection, leading to pneumonic plague, the form that can spread directly from person to person. But here's where it gets really interesting and kind of challenges older assumptions. Traditionally, the plague gene was thought to be absolutely essential for pneumonic plague. However, recent studies, particularly involving strains like Pistoides F, which naturally lack the plague gene, have shown that these play-lacking strains can still cause severe pneumonic plague, and they can even be transmitted person to person via aerosols without a flea vector. This suggests that Y. pestis has other compensatory virulence factors, alternative genetic tools if you like, that can step up when play is absent, allowing it to maintain its virulence. It just highlights the bacterium's remarkable adaptability and resilience.
So it's not a one-trick pony, even when a key tool is missing. That's a clever survival strategy. But how does it evade our immune system? The very defense is designed to fight off invaders, because that's what makes a pathogen truly successful.
Right, immune evasion is key, and Y. pestis has several sophisticated strategies. First, it produces the F1, or fraction 1 capsule. This is a protein-lipoprotein complex encoded by the PMT1 plasmid. Its primary function is to form this protective layer around the bacterium, making it much harder for our immune cells, specifically macrophages and other phagocytic cells, to engulf and destroy it. Essentially, the F1 capsule acts like a biological invisibility cloak, or maybe a slippery shield, allowing the bacteria to avoid detection and elimination.
And there's another one, the V antigen, right? It sounds like it works together with F1 somehow.
Correct. The V, or LCRV, antigens are encoded by the PCD1 plasmid, and they also contribute to preventing phagocytosis and modulating the host immune response. What's truly fascinating is how Y. pestis intelligently deploys these defenses based on its environment. It can actually survive and proliferate inside certain human white blood cells, specifically monocytes, but not neutrophils. It achieves this by producing these F1 and V antigens when it's at human body temperature, 37 degrees Celsius, which basically signals its presence within a mammalian host. Once it emerges from these monocytes, it is fully virulent, armed with these anti-phagocytic factors, and ready to cause widespread infection. In stark contrast, in the flea's cooler gut, these antigens aren't produced, which makes the bacteria susceptible to the flea's own immune responses. It's a remarkable example of temperature-dependent gene regulation.
That's a truly clever trick, hiding inside our own cells and activating its weapons only when needed. But for it to spread efficiently, it needs to get from the flea to us, and that involves something called biofilm formation. How does Y. pestis use biofilms to its advantage?
Biofilm formation. It's absolutely critical for efficient flea-borne transmission, especially for creating those massive human outbreaks. Y. pestis forms these dense, sticky biofilms in the flea's foregut, which can partially or completely block the gut. This blockage then causes the flea to become ravenously hungry and, critically, facilitates the regurgitation of a high dose of bacteria directly into the bloodstream of a new host during subsequent feeding attempts. This whole process is largely regulated by the Herm system, which orchestrates the production of an extracellular matrix, essentially creating a bacterial plug. Without this biofilm, the flea simply wouldn't be as effective a vector.
And like all living things, bacteria need resources, right? Just like us. What about iron uptake? Our bodies, as a defense mechanism, try to limit free iron to starve off invading pathogens. How does Y. pestis overcome that?
You're absolutely right. Iron is a critical nutrient for bacterial growth and proliferation, and the host environment is typically kept iron-limited precisely to hinder pathogens. Y. pestis has evolved incredibly highly efficient mechanisms for iron acquisition. It primarily uses the Yersinia-Bactin siderophore system. This involves small molecules that are secreted by the bacterium, bind to iron very tightly, and then transport it back into the bacterial cell. It can also acquire iron directly from heme-containing compounds like the hemoglobin in our blood, using other specialized transport systems. The PGM locus, which we've learned a lot about through genomic studies, is involved in both iron acquisition and storage. Without a functional PGM locus, the bacterium's virulence is significantly reduced, highlighting just how essential this ability to scavenge iron is for its survival and proliferation in the host.
And finally, a low calcium response. That sounds a bit counterintuitive. Why would a pathogen care about calcium levels, and how does it benefit from a low calcium response?
Yeah, the low calcium response, or LCR system, is actually a brilliant adaptive mechanism. It gets activated when Y. pestis finds itself in the flea gut, where calcium levels are naturally low compared to a mammalian host. This system, also encoded on the PCD1 plasmid, is crucial because it allows the bacterium to express a whole suite of additional virulence factors necessary for infection in a mammalian host later on. More importantly, it helps it evade the flea's immune system, allowing it to survive and multiply within its insect vector. Essentially, it's preparing it for subsequent transmission to a human or animal host. So it's a key adaptive mechanism for thriving within the flea, ensuring it's primed and ready for infection when it encounters a warm-blooded host.
So Y. pestis clearly has this remarkably sophisticated and adaptable genetic toolkit, but historically scientists also categorized it into different biovars. What exactly are those, and how do these traditional classifications connect to the history of the major pandemics?
Right, historically, microbiologists classified Y. pestis into three main biovars: Antiqua, Medievalis, and Orientalis. These divisions were based on relatively subtle phenotypic distinctions you could observe in a lab setting, like their ability to metabolize nitrate or ferment glycerol. The fascinating part, from a historical and epidemiological perspective, is how these biovars correlate so powerfully with the major plague pandemics we discussed earlier. The Antiqua biovar, with strains still found in persistent natural foci in Central Asia, Siberia, and parts of Russia, is believed to be linked to the Justinian plague, the first pandemic. Medievalis, primarily found in Central Asia, is now genetically confirmed to have descended from the bacteria that caused the second plague pandemic, which includes the Black Death. And finally, Orientalis strains, which originated in southern China around the late 19th century, are epidemiologically and genetically linked to the third plague pandemic and are the predominant biotypes circulating globally today. So these subtle phenotypic differences actually tell a profound story of the bacterium's historical journey, its diversification, and its recurring impact on humanity.
All right, let's shift gears now from the ancient scourge itself to the cutting-edge science that's helping us understand it. We're talking about reading the bones, how ancient DNA solved some of these long-standing historical mysteries. This sounds like archaeological detective work, but, with microscopes and sequencers. What's the biggest challenge when you're trying to extract information from something that's been buried for hundreds, maybe thousands of years?
The biggest challenge, and it's a truly significant one, is working with DNA that is incredibly degraded and often highly contaminated after centuries in the ground. I mean, imagine trying to piece together a shredded, ancient manuscript that's been buried for hundreds of years, exposed to the elements, and then trying to differentiate its ink from the soil and other microbial contaminants all around it. That's essentially the challenge with aDNA. The fragments are really short, often chemically damaged, and they're vastly outnumbered by DNA from the human host or environmental microbes. It's tough stuff.
So where do you even begin to look for this precious, degraded pathogen DNA? What are the best sort of time capsules for genetic material?
Well, the primary source for sample collection is often dental pulp and bone samples, usually from mass graves where plague victims were buried. But researchers particularly prefer teeth, and specifically the dental pulp located deep inside. The reason is that the dental pulp chamber offers a relatively sterile and remarkably protected environment, and the dried blood vessels within it provide a really high preservation potential for pathogen DNA. It's like a tiny, sealed biological time capsule within the tooth, protecting the DNA from environmental degradation much better than bone. For bone samples, the protocols involve meticulously removing surface contaminants and then pulverizing the bone under rigorously sterile conditions.
And I imagine preventing modern contamination is absolutely paramount, right? You don't want to find Y. pestis just because you accidentally introduced it from the lab environment or even from yourself. That would completely invalidate any findings.
Oh, absolutely correct. And this cannot be overstated. Strict protocols are absolutely essential to prevent modern DNA contamination. This means dedicated, ancient DNA facilities, completely separate from modern biology labs. Researchers wear full protective gear, head-to-toe suits, multiple layers of gloves, face masks. They use UV irradiation to sterilize all surfaces. They mechanically remove the outer surface of bones or teeth and pulverize samples, finally using aseptic techniques. Furthermore, negative controls are absolutely essential. You run blanks through every single step, from the initial extraction to PCR amplification, just to monitor for any tiny bit of laboratory contamination. It's a meticulous, almost surgical level of precision needed to ensure the authenticity of the ancient DNA.
Okay, so once you have these painstakingly collected and prepared samples, how do you even know if that elusive Y pestis DNA is present amidst all the human and environmental DNA? It sounds like finding a needle in a haystack, but the needle itself is mostly rust.
Right. So after DNA extraction, which typically uses highly efficient guanidinium silica-based methods, the initial screening involves a remarkably sensitive technique called quantitative PCR, or qPCR. This method targets a Y pestis-specific gene, usually the plague gene, which we talked about earlier. It's located on one of its virulence plasmids and is highly conserved within the species. If the plague gene is detected, even in tiny quantities, it's a strong preliminary indication that Y. pestis DNA is indeed present in the sample. For example, some studies might evaluate hundreds of DNA extracts from teeth, and perhaps only a quarter or maybe even a fifth of those yield a positive signal this way. It's really an initial filter to see if it's worth going further.
But to truly get into the intricate details of the pathogen's genetics, to build that evolutionary family tree, you need more than just a yes-no answer, right? You need to sequence the whole genome, or at least a significant part of it. How do you enrich for that tiny, precious bit of Y. pestis DNA amidst the overwhelming abundance of human and environmental DNA? That sounds like the real magic trick here.
That's where whole genome array capture comes in, and it's truly a key enabling technique for ancient pathogen genomics. It's really quite clever. Researchers custom design probes, which are short synthetic DNA sequences. They base these probes on the known chromosome of Y pseudotuberculosis. Remember, Y. pestis is a very close modern relative, and the known Y. pestis plasmids like PMT1 and PCD1. These probes are then used to capture or selectively pull out specific Y. pestis DNA fragments from the extracted mixture. It's much like using a magnet to pull out specific metal shavings from sand. This targeted enrichment allows for high-throughput sequencing, focusing the sequencing power and resources exclusively on the pathogen's genome, greatly reducing the noise from all the other DNA. This approach makes it possible to achieve decent genomic coverage from ancient samples, even with very small initial amounts of pathogen DNA. For instance, studies have achieved average genomic coverage of, say, 10.3x for a Barcelona sample, 19.3x for Bulgar City, and maybe 4.9x for Elvangen.
What do those numbers like 10.3x or 4.9x actually mean for us as listeners? Are they high, low, or just enough to paint a clear picture of the ancient genome?
That's an excellent question, because the raw numbers can be a bit abstract, can't they? They basically tell us about the resolution and the completeness of our genetic picture. For context, modern pathogen genomes are often sequenced at 30x coverage, or even more. So 10.3x means, on average, we've read each piece of the bacterial genome more than 10 times over. That gives us quite high confidence in the sequence we reconstruct. Even 4.9x, while lower, is often enough to piece together the critical parts of the genome and make robust phylogenetic comparisons, especially when you're looking for those single nucleotide differences, the SNPs. It's like having enough puzzle pieces, even if some are a bit worn or duplicated, to still see the full, intricate image of the past. It's pretty remarkable we can get this level of detail from samples that are centuries old.
So once you have all this raw genetic data, these millions of short DNA fragments, you need to make sense of it all. This is where bioinformatics, the sort of digital microscope, comes in. How crucial is this step for accurate analysis of such delicate, ancient DNA?
Bioinformatics is not just important. It's absolutely essential for accurate analysis and interpretation of aDNA data. You simply couldn't do it without it. Ancient DNA presents unique challenges beyond just degradation. Its fragments are typically very short, and there are specific postmortem damage patterns, like cytosine deamination, where cytosine bases chemically mutate into thymines over time. If you don't properly account for these issues, they can lead to serious misinterpretations and false positives in your results. Bioinformatics tools are specifically designed to computationally identify and correct for these hurdles, ensuring the data is as clean and reliable as possible before you draw conclusions.
What are some of the key pipelines or software used for this, these digital tools that piece together the past?
Yeah, there are several really sophisticated pipelines developed specifically for this field. Paleomics, for example, is known for being very user-friendly and offers toolkits for mapping these short ancient DNA reads to a reference genome, performing genotyping, and even taxonomic and metagenomic profiling of complex aDNA samples. It's built from the ground up to handle the complexities of degraded ancient DNA. Then there's EAGER, which stands for Efficient Ancient Genome Reconstruction. This is a really comprehensive pipeline. It includes modules for pre-processing raw data, meticulously mapping reads, analyzing DNA damage patterns, performing high-accuracy genotyping using advanced statistical tools, and even estimating contamination levels. Eager is notable for its graphical user interface, which makes it a bit more accessible for researchers who aren't deep bioinformatics experts. And finally, HOPS, or Heuristic Operational Pipeline System, is specifically tailored for detecting and authenticating pathogen DNA in complex metagenomic datasets, where the pathogen might be only a tiny fraction of the total DNA. It's known for its high sensitivity and specificity. These tools are really the unsung heroes of ancient genomics.
And these tools ultimately allow you to build something like an evolutionary family tree for the bacterium, right? To see how it branched and spread over time.
Precisely. The ultimate goal of these bioinformatics tools is to perform SNP analysis, identifying those single nucleotide polymorphisms, the single base differences in the DNA sequence, to build highly detailed phylogenetic trees. By meticulously comparing these SNPs across different ancient Y. pestis strains and comparing them to modern ones, scientists can reconstruct the evolutionary relationships and trace the historical journey of the pathogen across continents and centuries. For example, some seminal studies have considered over 3,300 or even 300 to 400 distinct SNP positions to build these intricate phylogenies. It's like creating a microscopic GPS tracking history of the plague, mapping its movements with incredible precision.
This is where it gets really interesting, because these scientific tools have truly rewritten the history books about the Black Death. One of the prevailing theories used to be that distinct plague lineages entered Europe via different pulses, or maybe that there were recurrent maritime imports from Asia fueling subsequent outbreaks. But the new evidence tells a dramatically different story, doesn't it? A single entry point that then just exploded.
That's right. The concept of multiple distinct plague lineages entering Europe in different ways, or continuous reintroductions from reservoirs outside Europe, was a significant and widely accepted hypothesis for a long time. However, recent groundbreaking studies, particularly those using this ancient DNA technology, have fundamentally challenged this view. What's truly fascinating is the comparison of a Y. pestis strain found in Barcelona, Spain, from the very early phase of the Black Death spring 1348. Remember, that was one of the main entry points into southern continental Europe, with a strain from London, where the earliest reports of the illness were in autumn 1348.
And what did that comparison reveal? Was it as different as we might have expected, given the distance and the few months difference?
The striking and quite unexpected finding was that the same strain of Y. pestis was present in both southern Europe, like Barcelona, and northern Europe, like London, within mere months of the Black Death's initial, incredibly rapid spread into the continent. And this wasn't just a similar strain, it was genetically almost identical, really remarkably little diversity. This strongly supports the notion of a single-wave entry of Y. pestis into Europe during the Black Death, characterized by surprisingly low genetic diversity in the pathogen during its early, incredibly rapid spread. This really reshapes our understanding, moving away from the multiple pulses model to one of a singular, incredibly potent wave that swept across the continent with shocking uniformity.
That's a huge shift in understanding how the Black Death swept across the continent. It tells us it wasn't a series of independent fires starting up, but a single, incredibly potent and genetically cohesive wave that just washed over Europe. But the story doesn't end there, does it? There's also a surprising revelation about plague's journey after the Black Death. The previous hypothesis suggested that Y. pestis might have traveled from Europe back toward Asia, eventually settling in China and maybe giving rise to the third pandemic. Did the new data support that rather counterintuitive idea?
Yeah, this is where the evidence from Bulgar City, in the Volga region of Russia, becomes incredibly significant. Researchers analyzed human remains from a plague burial in Bulgar City. This burial was dated by coin artifacts found with the bodies to sometime after 1362 AD, placing it squarely in the post-Black Death period. The genomic analysis of the Y. pestis strain from this site provided solid, pretty much undeniable evidence supporting that eastward migration model. The Bulgar City genome shared an additional unique genetic marker, a specific derived SNP position, with a London strain from the late 14th century. This strongly indicates that plague indeed traveled eastward from Europe subsequent to the Black Death. This was a really critical missing piece of the puzzle, connecting Europe back towards Asia.
So this isn't just a localized finding. It has global implications for understanding plague's entire history and its interconnectedness across continents. What does this eastward spread tell us about the broader picture?
Precisely. This eastward spread is now strongly connected to the origin of the 1-ion lineages that emerged later in the Yunnan province of China, and critically, to the 1-ARI strains. These 1-ARI strains were the ones responsible for the worldwide dissemination of plague during the third plague pandemic that started in the late 19th century, literally circumnavigating the globe. Furthermore, this also raises a compelling possibility regarding the 1-INT lineage, which today is primarily restricted to sub-Saharan Africa. Its shared ancestry with this Bulgar lineage suggests it might derive from a historical plague focus that existed somewhere along this eastern path. It could have potentially been introduced into Africa via European presence on the continent, perhaps through colonial trade routes or similar interactions. It's a complete reevaluation of the pathogen's global circulation, demonstrating how interconnected these historical events truly are, even across vast distances and times.
So, even after the initial devastating wave of the Black Death, Europe continued to experience recurrent plague outbreaks for centuries. This raised another critical question among historians and scientists. Were these later outbreaks due to constant, recurrent imports from Asia? Or was there potentially a long-term local European reservoir for the disease, maybe hidden away in rodent populations? What did the ancient DNA tell us about that ongoing debate?
This is an incredibly complex and frankly ongoing debate, with significant implications for understanding how diseases persist over long periods. To address this, researchers investigated a 16th century plague outbreak in Ellwangen, which is a relatively isolated city in southwestern Germany. They compared the Y. pestis strain found there to one from a London outbreak that occurred relatively soon after the Black Death, and also to the strain responsible for the much later Great Plague of Marseille, which struck in 1720-1722.
And what was the connection between Ellwangen and Marseille, given they were separated by so much time and geography?
Well, the Ellwangen strain was found to be directly ancestral to those from the Great Plague of Marseille. This indicates they shared a common genetic history and diverged from the same source population, even though centuries separated them. Critically, this lineage directly descends from the original Black Death strain that had circulated earlier in London and Barcelona. Now, the geographical location of Ellwangen, being more inland and seemingly part of a more restricted outbreak, combined with the timing, made introduction via external trade routes from outside Europe appear less likely for that specific 16th century event. It seemed much more plausible that the plague had been lingering somewhere closer to home within Europe itself.
So this evidence leans strongly towards a local European reservoir persisting for centuries, rather than constant reimportation from Asia. That's a significant finding, isn't it?
Yes, the data from Ellwangen strongly supports the idea of a local European reservoir for the disease, or possibly several reservoirs. This reservoir could have been comprised of small, perhaps isolated rodent foci, maybe in specific species like alpine marmots or other European rodent species not typically associated with human dwellings, which would have allowed the disease to persist locally without needing constant reintroduction from Asia. What's also notable is that the Ellwangen observance lineage, as it's been named, has no known extant descendants, none that we found anyway. This suggests that this specific European focus may no longer exist, and its extinction might have coincided with plague's sudden, rather perplexing disappearance from Europe in the 18th century, a historical event that's still debated by scholars.
And this ties into that popular theory about rats, doesn't it? The idea that a change in domestic rodent populations contributed to plague's disappearance. So the old nemesis, the black rat, was replaced by the brown rat.
It absolutely does, and it's a very compelling environmental factor to consider. The popular theory suggests an 18th century domestic rodent replacement. The black rat, Rattus rattus, which is a well-known harbinger of plague and thrives alongside humans, living in houses and attics, was largely replaced by the brown rat, Rattus norvegicus. Now, while brown Norway rats are also susceptible to plague, their ecological niche is different. They tend to live more in sewers and basements, and they have comparatively reduced contact with humans in domestic settings compared to the black rat. This biological shift in the dominant rat species could have significantly slowed or interrupted the transmission of the disease from any neighboring sylvatic or wild animal populations into human populations, contributing to its decline and eventual disappearance from Europe. It's a powerful example of how ecological changes can profoundly impact public health.
So it's not a simple single answer then, is it? It sounds like a confluence of factors, perhaps even a bit of luck, contributed to plague fading away in Europe.
No, it's highly nuanced and almost certainly far from a simple, singular explanation. The research indicates that a purely unidirectional dispersal of Y. pestis is unlikely. Multiple factors almost certainly contributed to its spread, its persistence for centuries, and its eventual disappearance. The epidemics in Germany and France, for instance, likely stem from just one of possibly several historical plague foci within Europe or its vicinity, It's entirely possible that there were concurrent plague foci harboring strains related to that Bulgar lineage we talked about, or the late 14th century London lineage, or even others we haven't yet identified through ancient DNA that were responsible for additional second plague pandemic outbreaks across Europe. The genetic data is really providing a much more complex and dynamic picture of plague's long and devastating history in Europe. It shows us this relentless evolving dance between the pathogen, the host, both human and animal, and the environment itself.
This is truly mind-blowing stuff. It's not just about tracking a pathogen through history, it's about understanding how such a devastating force might have actually reshaped our very bodies. Did the Black Death leave an indelible genetic mark on human immunity? Did it actually select for survivors?
Well, that's the profoundly fascinating idea here. Natural selection in action, on a truly dramatic scale. Epidemics as devastating as the Black Death, with their immense mortality rates and rapid onset, exert incredibly strong selective pressure on human populations. This means that individuals who happen to have even slight genetic advantages for survival in the face of the plague would be significantly more likely to survive, reproduce, and pass on those advantageous genes. This leads to shifts in allele frequencies over generations. It's literally evolution playing out in real time, albeit across many generations.
So, how did researchers investigate this monumental question? How do you even begin to look for genetic changes wrought by a medieval plague in people living today?
A groundbreaking study specifically focused on Ellwangen in Germany, bridging that historical gap quite elegantly. Researchers meticulously extracted DNA from 36 plague victims buried in a 16th century mass grave there. They then compared their genetic profiles to 50 modern-day inhabitants of Ellwangen. The key here was rigorously ensuring the genetic similarity of the populations over time, confirming that the modern inhabitants were indeed likely descendants of that historical population. This makes the comparison valid for detecting actual evolutionary changes in response to the plague, rather than just population migration or something else. This genealogical integrity was absolutely crucial.
And they didn't just look at random genes, right? They specifically targeted immunity-related ones, which makes perfect sense if you're looking for adaptation to disease.
Exactly. They developed a novel and highly sophisticated technique called in-solution hybridization capture to specifically enrich for and analyze a focused set of 488 human genes. These were genes known to be intimately related to immunity, including the highly important and very polymorphic HLA genes. This allowed for a very precise, almost surgical comparison, honing in on the genes most likely to have been impacted by such a severe infectious disease pressure.
So what were the key findings in these immunity-related genes? What specific genetic shifts did they discover that hint at our evolutionary battle with the plague?
Several intriguing and statistically significant changes were observed. Let's start with FcN2, or FcN2. Its function is to bind to bacteria, effectively acting as an early warning system. It triggers the complement pathway, a critical part of our innate immune system, to neutralize pathogens and promote their phagocytosis, their engulfment, by immune cells. Researchers found that two specific SNPs, a promoter variant and a coding change, were in complete linkage disequilibrium. That basically means they are almost always inherited together as a single functional unit, a haplotype. And this particular haplotype had increased significantly in frequency in the modern Ellwangen population compared to the 16th century plague victims.
So if I'm understanding correctly, it means these two genetic changes are almost always inherited together as a package. They don't typically get separated during recombination, which makes their increased frequency even more powerful evidence of selection acting on that package. And what does that mean for us today, having this increased frequency?
Precisely. Think of it as a combined genetic feature, a unified change that was strongly selected for over time. This specific FcN2 haplotype is associated with an increased serum concentration of the FcN2 protein and also subtly alters its binding properties. This makes it a very strong candidate for providing improved resistance to Y. pestis infection. It's a clear signal of adaptation, perhaps a genetic upgrade that likely helped individuals survive the plague and pass on that advantage to future generations, leading to its higher frequency today.
And another one you mentioned was NLRP14. What about that gene, and what was its evolutionary story in this context?
Right, NLRP14 is another fascinating gene. It belongs to a group of proteins called inflammasome complexes, which are basically intracellular pattern recognition receptors. They play a critical role in triggering local and systemic inflammatory responses to microbial invasion and can even initiate the self-destruction of infected cells, a process called pyroptosis, to contain infection. Inflammasomes are known to be important in the immune response to Yersinia infection specifically. The study found a specific coding change variant in NLRP14 specifically, RS108397008, which changes an amino acid, a glutamic acid, to a lysine at position 808. This variant was found at a lower frequency in modern individuals compared to the plague victims from the 16th century.
So a decrease implies it might have been detrimental to have that variant during the plague. An evolutionary battle, you called it, where certain traits were actively selected against.
That's the compelling implication, yes. This coding change occurs in a domain that's known to control ligand specificity in similar molecules. That means it likely affects how NLRP14 recognizes components of Y. pestis. This evolutionary battle with the pathogen suggests that the variant that is now less frequent might have been less effective at mounting a protective response, or perhaps even detrimental during the infection. Maybe it caused an overzealous immune response that led to more collateral damage to the host rather than effective pathogen clearance. It suggests the plague wasn't just killing the weak, perhaps, but actively selecting against certain types of immune responses.
Now, what about some of the genes that have been previously suggested in other studies to be protective against plague, like CCR532? That's the one famous for also offering a degree of resistance to HIV. Did that show a significant change in the Elvongan population?
Interestingly, and somewhat counterintuitively based on some prior assumptions, the study found no significant frequency differences for CCR532, nor for specific variants in TLR4, another immune gene previously associated with plague resistance in some contexts. This challenges some of the earlier assumptions and existing hypotheses in the field. It suggests that while these genes certainly have important immune functions and might be relevant in other contexts or perhaps other populations, they may not have played a significant role in providing protection against Y. pestis during the Black Death in this specific Elvongan population they studied. This is exactly why replication and studies in diverse populations are so crucial in genetics.
So, beyond those specific genes, the HLA system is this massive, incredibly complex part of our adaptive immune response. It's known to be remarkably diverse between people. Did they find shifts there, given its absolutely central role in fighting off pathogens?
Oh yes. What's profoundly fascinating here is that HLA, or human leukocyte antigen, molecules are encoded by the most polymorphic gene complex in humans. They are absolutely crucial for initiating and directing our adaptive immune response to infection. They work by presenting pathogen-derived peptides, little bits of the invader, to our T cells, essentially signaling to the immune system exactly what to attack. Because of this central role, significant shifts in HLA allele frequencies are a very strong, almost undeniable indicator of natural selection at play. It's like finding a genetic fingerprint left behind by an intense evolutionary pressure event like a major pandemic.
And were there specific HLA alleles that changed dramatically in frequency, either increasing or decreasing over the centuries between the plague victims and modern people?
Yes, very clear and statistically significant shifts were observed. One notable increase was in an allele called HLA-DRB113. Its frequency increased significantly in modern individuals. It soared from about 5.6% in the 16th century victims to a striking 17.0% in the modern Elvongan population. What's particularly compelling is that this allele is also associated with increased resistance to Mycobacterium tuberculosis, the bacterium that causes TB. Like Y. pestis, M. tuberculosis can invade and survive within macrophages. This suggests a potential shared adaptive immune pathway, maybe a common cellular defense mechanism that could offer resistance to both pathogens, indicating a potentially broad immune advantage conferred by this allele. And what about the ones that decreased? Were certain HLA types becoming less common over time, suggesting they might have been disadvantageous? Conversely, yes. Two alleles, HLA-B51.01 and HLA-C06.02, both decreased significantly in frequency in the modern population compared to the 16th century victims. HLA-B51.01 went from a substantial 15.3% in the 16th century down to just 6.0% today, and HLA-C06.02 dropped from 13.9% down to 5.0%. These are not minor shifts, they are really quite traumatic changes. The statistical simulations performed by the researchers strongly suggest that natural selection was the driving force behind these changes. The estimated selection coefficients for these shifts, for instance, -0.25 for B51.01 and -0.27 for C06.02, negative indicating selection against, and a positive 0.37 for DRB113.01, indicating selection for, fall well within ranges previously reported for highly selected MHC genes. Imagine a genetic lottery where the stakes were literally life and death. These coefficients tell us just how profoundly those particular gene variants influenced survival during the plague era.
And the HLA system also interacts with other parts of the immune system, like natural killer cells, through something called KLIR receptors. Was there a connection found there, suggesting a more complex interplay of innate and adaptive immunity?
That's right. It raises an important question regarding the innate immune system's frontline defense. HLA class IL alleles interact with killer cell immunoglobulin-like receptors, or KIRs, which are found on natural killer, or NK, cells. These interactions modulate innate immunity and provide a rapid, non-specific defense against infected or stressed cells. The study found a higher incidence of the combination of KIR3DL1, which is an inhibitory receptor, and HLA-B allotypes that encode an amino acid, isoleucine, at position 80. This combination is often referred to as I80 plus SPIC-B. This specific combination permits the strongest inhibitory interaction between KIR3DL1 and HLA-B, and it was found to be more frequent in 16th century plague victims, present in 53% of those individuals compared to only 26% in modern Ellingen individuals.
What's the interpretation there? Could that combination have actually been a disadvantage during the plague, maybe leading to a counterproductive immune response?
That's the compelling interpretation put forth by the researchers, yes. They suggest that this specific, strong inhibitory combination might have been disadvantageous for individuals infected with Y. pestis. It could potentially lead to what's described as excess collateral damage caused by NK cells in response to the infection. In essence, an overzealous or perhaps misdirected innate immune response, maybe due to the strong inhibition, might have harmed the host more than it effectively cleared the pathogen, leading to poorer outcomes. This points to a really complex interplay where certain immune strengths can, paradoxically, become liabilities in the face of specific pathogens. It's a fine line between protection and self-harm.
So these findings are incredibly significant, providing compelling, maybe the first real direct evidence of human genetic adaptation to such a devastating historical event. But there's always nuance in science, isn't there? It's rarely a simple definitive answer right off the bat.
Absolutely. And the researchers themselves, with appropriate scientific rigor, acknowledge that some of the frequency changes they observed, while biologically intriguing and suggestive, do not withstand multiple testing correction. This means that while they are powerful leads and very suggestive, more extensive studies and replication in diverse geographic contexts are definitely needed to confirm their statistical significance with absolute certainty. It really highlights the ongoing iterative nature of scientific research. These findings are considered first evidence for evolutionary adaptive processes in this context, and they specifically require replication in different geographic settings and also functional tests in the lab to definitively confirm the specific links to Y. pestis resistance and the precise mechanisms at play.
And this is a critical point. Did the evolution of human immunity actually drive the reduction of Y. pestis virulence over time, or maybe its eventual disappearance from Europe? Because that would be an incredible testament to human resilience if our own bodies fought back evolutionarily.
While this study's findings do not provide support for the proposition that evolution of human immunity drove reduction of Y. pestis virulence and its disappearance from Europe. While they show clear signs of human adaptation to the plague, that our genes were indeed shaped by this selective pressure, the disappearance of the pathogen itself from Europe was likely due to other complex factors. Maybe environmental changes, maybe shifts in host populations like the rat dynamics we discussed, or perhaps even the gradual development of better public health practices and quarantine measures. It also leaves open the intriguing possibility that other massive epidemic challenges that happened since the 16th century, such as smallpox or tuberculosis, could have also exerted selective pressure on these same immunity genes, complicating the picture. Although, importantly, no evidence of these other pathogens was found in the plague victims' DNA in this particular study. It's just a continuous evolving story between pathogens and humanity, where both sides are constantly adapting and influencing each other.
So we've peered deep into the past, uncovered ancient genetic mysteries, and seen how a devastating plague might have shaped our own DNA. But it's so important to remember that Yersinia pestis isn't just a relic of history, is it? It's not just a ghost from a distant past. It's still very much with us today.
That's a crucial point that we really must emphasize. Yersinia pestis is still a significant public health threat today, causing sporadic infections and occasionally local epidemics across the globe. The organism persists in what are called natural foci. These are stable, long-term reservoirs within wild rodent populations across vast regions of Asia, Eurasia, Africa, and also the Americas, including parts of the western United States. These natural reservoirs, spread across so many different ecosystems, make it practically impossible to completely eradicate the pathogen from nature. It's simply part of the global ecosystem now, unfortunately.
And how is it primarily transmitted in a modern context? Is it still mostly the fleas and rats we often hear about from history, or are there new routes of concern we need to be aware of?
Well, the primary route of transmission to humans still remains flea-borne, from infected wild rodents to humans. While direct human contact with these wild rodents might be relatively rare for most people, flea bites during such encounters, or even from fleas leaving a dead rodent, can readily transmit the plague. Then there's secondary transmission, which is human to human. This occurs via respiratory droplets from pneumonic plague, where the infection has established itself in the lungs and becomes highly contagious through coughing or sneezing. People can also become infected, though less commonly, from ingesting contaminated animal products or handling the tissues of infected animals, for example. Hunters skinning an infected rabbit or coyote, or even sometimes pets bringing infected fleas into the home. While urban plague has been largely controlled in many communities by improved hygiene and effective rat control, sylvatic plague, the kind that circulates among wild mammals, remains a persistent challenge and is very difficult to eliminate due to the widespread distribution of its mammalian reservoirs and flea vectors.
So, if someone were unfortunate enough to contract plague today, how would it typically present clinically? And how do modern doctors diagnose and, crucially, treat it? What are the symptoms people should be aware of, particularly if they live in or travel to areas where it exists?
Right, the clinical presentation can vary depending on the route of infection. The most common initial form is bubonic plague. This affects the lymph nodes, leading to those characteristic painful swollen lymph nodes called buboes, typically in the groin, armpit, or neck, closest to where the flea bite occurred. This is usually accompanied by a sudden onset of high fever, chills, headaches, profound weakness, often nausea, and sometimes vomiting. The incubation period is usually under 7 days after a flea bite. Importantly, human-to-human transmission is very rare with this bubonic form. Now, if the Y. pestis bacteria manage to escape the lymph nodes and enter the bloodstream directly or spread from the buboes, it leads to septicemic plague. This can cause more systemic symptoms, sometimes without obvious buboes, including significant gastrointestinal distress like severe nausea, vomiting, diarrhea, and abdominal pain, along with fever and signs of shock. The most concerning form, from a public health perspective because of its transmissibility, is pneumonic plague. This presents with a very rapid onset of high fever, headache, overwhelming weakness, and quickly progressing pneumonia. Symptoms include severe shortness of breath, chest pain, and a productive cough, which may contain blood or be watery. This is the form that is transmitted human to human via aerosols, making it highly contagious and requiring immediate isolation and treatment.
And do the specific virulence factors we discussed earlier, things like the F1 capsule or the pleioprotease, do they play a direct role in how these different symptoms manifest? Do they literally shape the disease we see in a patient?
They absolutely do. They really act as the orchestrators of the clinical manifestations you see. For instance, the F1 capsule protein, by inhibiting phagocytosis and allowing the bacteria to multiply relatively unchecked in the lymph nodes, directly contributes to those characteristic painful buboes seen in bubonic plague. The pleioprotease, or plasminogen activator, is thought to drive the rapid signs of sepsis, often seen in septicemic plague, by degrading fibrin clots and other extracellular matrix components. This facilitates the bacteria's unchecked dissemination throughout the bloodstream. And both the pleioprotease and the type 3 secretion system, or T3SS, which injects bacterial proteins directly into host cells to manipulate them, are deeply implicated in the severe respiratory symptoms of pneumonic plague. They disrupt cellular functions, promote massive inflammation, and cause extensive tissue damage in the lungs. Understanding these virulence factors is crucial not just for academic knowledge, but for developing targeted diagnostics and more effective treatments down the line.
So, how do labs actually confirm a plague diagnosis, especially given how dangerous the samples can be and the strict biosafety levels required? What's the process?
Confirmation is typically done through a combination of techniques. There are classical bacteriological methods like gram stain and culture from a bubo aspirate, drawing fluid from the bubo, or from blood or sputum if pneumonic plague is suspected. There's also serological examination to detect antibodies against Y. pestis, though that takes time to develop. And increasingly, there are rapid diagnostic tests, RDTs, and highly specific PCR tests looking for bacterial DNA. Due to the high risk of severe infections for laboratory personnel, especially from aerosols generated during testing, samples potentially containing Y. pestis must be handled with extreme caution and in very specific environments. Immunological tests and nucleic acid-based detection methods generally require a biosafety level 2 or BSL-2 cabinet. However, any manipulation of live cultures that could produce aerosols or involves large volumes or concentrations of bacteria absolutely requires a biosafety level 3 or BSL-3 environment for maximum safety.
And just to give our listeners a clear picture, what's the practical difference between working in a BSL-2 and a BSL-3 lab when handling something as potentially deadly as Yersinia pestis?
That's a vital distinction. In a BSL-2 lab, you're typically working inside a contained biological safety cabinet, wearing standard personal protective equipment like gloves, a lab coat, and eye protection. The focus is on limiting direct contact and containing splashes or minor aerosols. But for a BSL-3 lab, it's a whole other level of precaution. You have specialized negative pressure ventilation systems to ensure air flows into the lab, not out, preventing airborne escape. There are restricted, often double-door entry systems acting like airlocks. And researchers often wear much more extensive protective gear, sometimes including respirators or even powered air purifying respirators, PPRs, especially when working with cultures. It's about ensuring absolute containment and preventing any possible airborne transmission, acknowledging the severe risk of pneumonic plague if aerosols are generated. There are also challenges just in identifying it correctly sometimes. Conventional biochemical identification systems and even advanced methods like Malditos mass spectrometry can occasionally misidentify Y. pestis, potentially confusing it with its less virulent cousin, Y. pseudotuberculosis, or even other related bacteria. Therefore, any initial identification is usually considered presumptive until it's rigorously confirmed by a specialized reference laboratory. Specific tests often include detecting the F1 antigen using immunochromatographic assays, like those rapid dipstick tests for quick on-site detection, or maybe ELISA in the lab. PCR tests are very common now, targeting specific genes like CAF1, which encodes the F1 antigen, or PLAY. But it's important to remember that play-based detection can be unreliable in regions where those play-lacking strains we discussed earlier might be common, highlighting a need for using multiple diagnostic targets for confirmation.
What about treatment then? Is it still as deadly if left untreated today as it was during the Black Death?
Thankfully, no, absolutely not. This is where modern medicine, specifically antibiotics, has made an enormous difference. Bubonic plague, the most common form, typically responds very quickly and effectively to appropriate antibiotic therapy. Treatment dramatically lowers the mortality rate from about 60% or even higher if left untreated, down to as low as 5% or potentially even less if treatment is started promptly within the first few days of illness. Oral antibiotics like doxycycline and ciprofloxacin are generally recommended as highly effective first-line treatments. However, a significant and worrying development is the emergence of multi-drug resistant, or MDR, strains. An isolated MDR strain was identified in Madagascar a few years ago, for instance, resistant to a whole range of critical antibiotics, including streptomycin, chloramphenicol, and tetracycline. This raises serious public health concerns, and given Y. pestis' classification as a Category A bioterrorism agent, even potential bioterrorism threats. In such scenarios, careful selection of effective antibiotics based on susceptibility testing becomes absolutely paramount, and research into new therapeutics and alternative treatments is ongoing and vital.
So, what's being done globally to prevent future outbreaks? And what's the current state of vaccine development? Is there a light at the end of the tunnel for completely eradicating this ancient foe someday?
Well, continuous preventive surveillance for plague and other zoonotic diseases is absolutely necessary and is a global effort, coordinated by organizations like the WHO and national health agencies. This involves active monitoring of wild animal populations, especially rodents, in known endemic areas for signs of the pathogen, testing fleas, and ensuring rapid response capabilities for any detected human cases or animal die-offs. While advancements are certainly being made in plague vaccine development, including research into oral vaccines, bacteriophage-based therapies, and new recombinant vaccines designed to be more effective and have fewer side effects than older kill vaccines, there are still ongoing challenges. Developing a vaccine that provides long-lasting, robust protection against all forms of plague, including pneumonic, remains difficult. More research and investment are definitely needed to develop universally effective, safe, and accessible vaccines for widespread use, particularly for high-risk groups or in outbreak situations. But the hard truth is, the enduring presence of Y. pestis in these vast natural foci across the globe means we probably cannot eradicate it entirely from the environment, at least not with current technology. So vigilance, rapid diagnosis, effective treatment strategies, public education in endemic areas, and ongoing research remain our primary, critical defenses against this ancient, yet still very real public health threat.
Wow, what an incredible journey we've taken today. From the chilling historical impact of the Black Death to the cutting-edge science that's literally reading history, written in bones and genes. We've seen how ancient DNA has not only confirmed old suspicions, but also revealed the global pathways of ancient plagues, really challenging long-held assumptions about their spread with stunning new evidence.
And we've also unpacked the subtle but incredibly significant shifts in human genes that show our evolutionary dance with this formidable pathogen. It's not just about what happened, but really how and why. Uncovering these hidden stories within both the pathogen itself and the very fabric of our own bodies, showcasing humanity's remarkable, sometimes costly resilience.
It truly reinforces the idea that scientific advancements allow us to understand so much more about our past, connecting it directly to our present. Yersinia pestis isn't just some relic in a dusty textbook. Its enduring presence today highlights the critical importance of understanding infectious diseases, not just for historical context, but for ongoing public health and really our collective future.
Yeah, if we connect this to the bigger picture, our deep dive into why pestis reveals this continuous, evolving story between pathogens and humanity. It's a relentless back and forth of adaptation on both sides. And this raises an important, perhaps even a provocative question for all of us. What unexpected evolutionary pressures might today's rapid environmental changes or our unprecedented global interactions and travel be exerting on our genes right now? Are they preparing us or maybe challenging us for tomorrow's unseen threats?
Food for thought, indeed, and a powerful reminder that the story of our species is just inextricably linked to the microscopic world around us. Always has been, always will be. We encourage you, our listener, to continue exploring these fascinating intersections of history, biology, and human resilience. Until next time, keep digging deeper.